Hello students, the topic of today's module is photophosphorylation. In this module, we'll be learning about photophosphorylation, types of photophosphorylation and its comparison to mitochondrial oxidative phosphorylation. So what is photophosphorylation? Photophosphorylation can be defined as a process of synthesizing energy-rich ATP molecules from ADP and inorganic phosphates in the presence of light. Chloroplasts and photosynthetic bacteria obtain high-energy electrons by means of photosystems that capture the electrons that are excited when sunlight is absorbed by chlorophyll molecules. The combined activities of the two plant photosystems move electrons from water to NADP+, conserving some of the energy of absorbed light as NADPH. Simultaneously, protons are pumped across the thylakoid membrane and energy is conserved as an electrochemical potential, and the proton gradient is used to drive the synthesis of ATP. In 1954, Daniel Arnon and his colleagues discovered that ATP is generated from ADP and inorganic phosphate during photosynthetic electron transfer in illuminated spinach chloroplasts. Support for these findings came from the work of Albert Frankel, who detected light-dependent ATP production in pigment-containing membranous structures called chromatophores, derived from photosynthetic bacteria. Investigators concluded that some of the light energy captured by the photosynthetic systems of these organisms is transformed into the phosphate bond energy of ATP. This process is called photophosphorylation to distinguish it from the oxidative phosphorylation in respiring mitochondria. Then in 1966, Andre Jagendorf showed that chloroplasts synthesize ATP in the dark when an artificial pH gradient is imposed across the thylakoid membrane. To create this transient pH gradient, he soaked chloroplasts in a pH 4 buffer for several hours and then rapidly mixed them with a pH 8 buffer containing ADP and inorganic phosphate. The pH of the stroma suddenly increased to 8, whereas the pH of the thylakoid space remained at 4. A burst of ATP synthesis then accompanied the disappearance of the pH gradient across the thylakoid membrane. Now, this experiment was one of the first to support the chemiosmotic hypothesis put forward by Peter Mitchell that ATP synthesis is driven by proton motive force. During photosynthesis, light induces electron transfer through photosystems 2 and 1 and the cytochrome Bf complex. At various stages in this process, protons are released into the thylakoid lumen or taken up from the stroma, generating a proton gradient. The gradient is maintained because the thylakoid membrane is essentially impermeable to protons. The thylakoid space becomes distinctly acidic with a pH approaching 4. The light-induced transmembrane proton gradient is about 3.5 pH units. The energy which is inherent in the proton gradient, called the proton motive force, is described as a sum of two components, a charge gradient and a chemical gradient. In chloroplasts, nearly all of the proton motive force arises from the pH gradient. Now let's look at the ATP synthase of chloroplasts. The ATP synthase of chloroplasts closely resembles those of mitochondria and prokaryotes. The proton motive force generated by the light reactions is converted into ATP by the ATP synthase of chloroplasts, also called the CF1-CF0 complex. C stands for chloroplasts and F for factor. CF1-CFO ATP synthase closely resembles the F1-F0 complex of mitochondria. CF0 conducts protons across the thylakoid membrane, whereas CF1 catalyzes the formation of ATP from ADP and inorganic phosphate. The CF node subunit is embedded in the thylakoid membrane. The membrane orientation of CF1 CF node is reversed compared with that of the mitochondrial ATP synthase. However, the functional orientation of the two synthases is identical. Protons flow from the lumen through the enzyme to the stroma or matrix where ATP is synthesized. Because CF1 is on the stromal surface of the thylakoid membrane, the newly synthesized ATP is released directly into the stromal space. Likewise, NADPH formed by photosystem 1 is released into the stromal space. Thus, ATP and NADPH, which the products of the light reactions of photosynthesis, are appropriately positioned for the dark reactions which will take place subsequently in the stroma, where carbon dioxide is converted into carbohydrate. There are two types of photophosphorylation: non-cyclic photophosphorylation and cyclic photophosphorylation, based on the path the excited electrons take during the electron transfer process in the photosystems. Photosynthesis in plants and cyanobacteria produces both ATP and NADPH directly by a two-step process called non-cyclic photophosphorylation. Because two photosystems called photosystem 1 and 2 are used in a series to energize an electron, the electron can be transferred all the way from water to NADPH. As the high-energy electrons pass through the coupled photosystems to generate NADPH, some of their energy is siphoned off for ATP synthesis. PS2 splits water and converts it to oxygen, whereas only PS1 transfers electrons to the final electron acceptor, which is NADP+, and reduces it to NADPH. The cytochrome Bf complex helps in the transport of excited electrons from PS2 to PS1 while pumping protons across the thylakoid membrane into the thylakoid space. Here, the path of the electrons is unidirectional and non-cyclic, and therefore this process is called non-cyclic photophosphorylation. So, in non-cyclic photophosphorylation, there is involvement of both the photosystems 2 and 1. Splitting of water molecules takes place. There is formation of molecular oxygen as a byproduct, and there is concomitant production of ATP and NADPH. Next, we'll study the cyclic photophosphorylation. In the non-cyclic photophosphorylation, we just discussed, high-energy electrons leaving the photosystem 2 are harnessed to generate ATP and are passed on to the photosystem 1 to drive the production of NADPH. This produces slightly more than one molecule of ATP for every pair of electrons that passes from water to NADP+ to generate a molecule of NADPH. But 1.5 molecules of ATP per NADPH are needed for carbon fixation. So to produce extra ATP, the chloroplasts of some species of plants can switch photosystem 1 to a cyclic mode so that it produces ATP instead of NADPH. This process is called cyclic photophosphorylation. Here, the high-energy electrons from photosystem 1 are transferred to the cytochrome Bf complex rather than being passed on to NADP+. And from the cytochrome Bf complex, the electrons are passed back to the photosystem 1 at a low energy. So, besides the conversion of some light energy to heat, the proton is pumped across the cytochrome Bf complex as the electrons pass through it again, thereby further increasing the electrochemical proton gradient across the thylakoid membrane. This proton motive force then drives the ATP synthase for ATP production. This process is analogous to that in purple non-sulfur bacteria. Also, on some occasions, when the ratio of NADPH to NADP plus is very high, NADP plus may be unavailable to accept electrons from reduced ferredoxin. In this case, electrons arising from the reaction center of photosystem 1 may take this cyclic pathway as an alternative pathway that does not end at NADPH. The electron in reduced ferredoxin is transferred to the cytochrome BF complex rather than to NADP+. This electron then flows back through the cytochrome BF complex to reduce plastocyanin, which can then be reoxidized by P700+ in PS1 to complete a cycle. So, as mentioned before, the net outcome of this cyclic flow of electrons is the pumping of protons by the cytochrome BF complex. So, in cyclic photophosphorylation, the electrons take a cyclic path involving only photosystem 1. Photosystem 2 is not involved. There is production of ATP and no concomitant production of NADPH. There is no production of molecular oxygen. The relative activities of cyclic and non-cyclic electron flows can be regulated by the cell to determine how much light energy is converted into reducing power in the form of NADPH and how much into high-energy phosphate bonds in the form of ATP. Now, let's compare photophosphorylation and oxidative phosphorylation in mitochondria. Several properties of photosynthetic electron transfer and photophosphorylation in chloroplasts indicate that a proton gradient plays the same role as in mitochondrial oxidative phosphorylation. The principles of ATP synthesis in chloroplasts are nearly identical with those in mitochondria. ATP formation is driven by a proton motive force in both photophosphorylation and oxidative phosphorylation. Number 1. The reaction centers, electron carriers, and ATP forming enzymes are located in a proton impermeable membrane, that is the thylakoid membrane, which must be intact to support photophosphorylation, just as the electron transport chain components are located in the inner mitochondrial membrane. Number 2. Photophosphorylation can be uncoupled from electron flow by reagents that promote the passage of protons through the thylakoid membrane. Number 3. Photophosphorylation can be blocked by benturicidin and similar agents that inhibit the formation of ATP from ADP and inorganic phosphate by the mitochondrial ATP synthase. Number 4. ATP synthesis is catalyzed by F1-F0 complexes located on the outer surface of the thylakoid membranes that are very similar in structure and function to the F1-F0 complexes of mitochondria. However, in chloroplasts, nearly all of the proton motive force arises from the pH gradient, whereas in mitochondria, the contribution from the membrane potential is larger. The reason for this difference is that the thylakoid membrane is quite permeable to chloride and magnesium ions. The light-induced transfer of protons into the thylakoid space is accompanied by the transfer of either chloride in the same direction or magnesium ions, like 1 magnesium ion per 2 protons, in the opposite direction. Consequently, electrical neutrality is maintained and no membrane potential is generated. A pH gradient of 3.5 units across a thylakoid membrane corresponds to a proton motive force of 0.20 volts. For both mitochondria and chloroplasts, the catalytic site of ATP synthase is at a pH of about 8, and is located in a large organelle compartment, that is the inner matrix in mitochondria and the stroma in case of chloroplasts, where it is packed full of enzymes. So that's all we'll be learning for today. Let us summarize what we have learned today. Photophosphorylation is the process of synthesizing energy-rich ATP molecules from ADP and inorganic phosphate in the presence of light. Here, the proton motive force generated by the light reactions during photosynthesis is converted into ATP by the ATP synthase in the thylakoid membranes of chloroplasts. The ATP synthase of chloroplasts closely resembles the ATP synthesizing assemblies of bacteria and mitochondria. There are two types of photophosphorylation, cyclic and non-cyclic, depending on the part of the high energy electrons take during the electron transfer process in photosystems during light reaction of photosynthesis. During non-cyclic photophosphorylation, the electrons take a linear path when they are being transferred from water to NADP+, involving both photosystems 2 and 1. There is splitting of water molecules with the liberation of molecular oxygen as a byproduct and concomitant production of both ATP and NADPH. And during cyclic photophosphorylation, the electrons take a cyclic path. Photosystem 2 is not involved, and the electrons evolving from PS1 are transferred back to PS1 via cytochrome Bf complex. So there is no production of NADPH or oxygen. And lastly, the principles and mechanism of photosynthetic electron transfer and photophosphorylation in chloroplasts clearly resemble the mitochondrial oxidative phosphorylation. Thank you.